Thank you very much for the kind introduction. So I have to admit, I did struggle with this preparation because I've never attended this conference before. And the first two speakers really scared me. They're so wonderful. And their research is kind of, I don't know, 100 levels above mine. So my lab at the University of Maryland is very small. I have two people in my lab. I have one PhD student who's graduating in a few months, and then I have one technician. That's it. So our research has been really, really focused since I joined UMB. So I joined in 2022 as introduced. Um, so I did some metabolism. I actually my AHA award was on metabolism. By the regulation by of metabolism by TEAT1, that's a hippo pathway downstream target. So I have a question to the audience. Who does heart research? Okay, not too many. Great, wonderful. Okay. So I think so yeah, I did not really bring data today. So because it's actually not super like my current research is not super relevant to metabolism. As I've said, I did quite some, the traditional seahorse, Ouroboros, all kinds of metabolic experiments back when I was a trainee. And anyhow, when I started my lab, we just kind of worked on a very novel regulator. And then surprisingly, we found that it regulates cardiomyocyte proliferation. And therefore, I just kind of went with the flow and then just started to uh, study the proliferation. So this would hopefully you would just learn something about maybe development of cardiomyocyte from my talk, and hopefully it's you're gonna it's gonna be a lighter session with no data. But I'll talk about my data. I'll just interject when appropriate. So okay. So when Zion invited me, he said, "Can you talk about cardiac plasticity?" I heard this term a lot, but I still had to Google it, what it means. Um, so essentially, these are all the interchangeable terms: flexibility, remodeling, adaptability. Basically, plasticity is just this, the ability to change. Um, and then how it might be related to proliferation, whatever I'm going to present today. Oh, okay. The mouse works. Okay. Cool, cool. All right. Thank you. Thank you. So, um, so who studies proliferation here? Nobody. Okay, good. Then I can just talk whatever. So, so why that, so as I said, I was really struggling because I haven't really followed up with metabolism articles for a while. So when I looked up, I found a few articles, review articles from, say, Dale Label, the big metabolism person, and then some other people. I was like, I really have not followed up for a long time. So I was really not super comfortable presenting that topic, but I do want you, after this talk, I do want you to learn that proliferation is also related to metabolism, and all these are kind of um, related to plasticity. So first of all, um, when we compare the embryonic cardiomyocytes versus the adult cardiomyocytes, um, they prefer to use different types of fuel. This is the first difference. The embryonic cardiomyocytes, they prefer using glucose, and they prefer using the glycolysis pathway, and they produce lower ATP because they just don't need as much. The structure of embryonic cardiomyocytes, they're much less complicated. They have much less sarcomere. They have much less mitochondria. However, in adults, they prefer to use oxidative phosphorylation, and they can produce higher ATP. So this um, is, let's say, um, the first difference. And then in terms of cardiac plasticity in adults, um, in this very nice review article written by Joe Hill and I forget the second person, but they published this review article on cardiac plasticity almost 20 years ago, talking about how the hearts adapted to environmental changes. So as we know, physiological hypertrophy happens if we exercise a lot, and then also during pregnancy. And then the pathological adaptations such as atrophy, like such, for example, prolonged bed rest, or people with cancer, or in the space when they're weightlessness, and the heart would go through atrophy. However, in more hypertensive status, myocardial infarction, or super activated, say beta adrenergic activation, the hearts would go through pathologic hypertrophy. And then with prolonged stress, they would start to develop heart failure, and then also the arrhythmia. So these are kind of known for just as long as we know. And then so it brings up to the two aspects that I'm going to talk about today. One is the cardiomyocyte development and regeneration, and then the second is metabolism and heart failure. Um, all right, so again, I'm just trying to cue the word plasticity. So first of all, developmental plasticity, as we know, we learned from biology book, the heart would go through hypertrophy, hyperplasia first, and then hypertrophy. So here I'm just comparing three different species. One is zebrafish, second is murine, mice rats, and then the third is human and sheep. So these three different species, they have different types of regeneration capacity. Say zebrafish, they can all their heart can always proliferate. And then for murine, say mouse and rats, their cardiomyocyte would exit cell cycle by day 7. And then after day 7, they would start to just grow bigger, and meaning enter into the hypertrophy session. And then in human, this is very similar. However, it might be a little bit different timeline, but very similar. So this is the developmental differences. And then I'm not sure, have any of you asked why? Like why the fish can regenerate, but we cannot, but the mammals cannot? No. Or maybe you may be shy to answer, but um, so I was never a developmental person, but then, however, my grant got into developmental biology section. I was like, no, it doesn't fit. I fought with NIH, but they were like, no, yours fits. But anyhow, the point is, I started to learn about developmental biology, and then I figured it could really help us learning about how that we can help developing therapeutics. Um, okay, I'm getting into more of the details. So the cardiomyocyte during development, there will be, so in mice, let's say in mice, there will be two DNA synthesis peaks. The first peak would be during mid gestation, and then the second peak would be very soon after birth, between day 3 and between day 4 and day 6. So basically, you can imagine those two DNA synthesis peaks, and then after day 7, they was just kind of start to grow bigger. But then one thing I did not know until I studied proliferation myself is that they actually still proliferate, but then they proliferate in another sense, as you can see from the right panel. They undergo this endomitotic cell cycle, meaning they're proliferating within the cycle. They have this karyokinesis, meaning the nuclei would just split. However, the cell does not. There's no cytokinesis. So why is this biologically important? Because I think people study liver, they all also know this liver cells, they also have these polyploidization, and also cancer cells. Basically, evolutionary people think this just was trying, the cells were trying to make themselves stronger. Um, However, the trade-off is that it's harder for the cells to split, because they just don't have those proliferative machinery in their genes. They just kind of, you can imagine, they kind of use all of their energy trying to perform some function. And then the zebrafish, as I mentioned earlier, so adult zebrafish, they can always regenerate their heart, and they can fully regenerate their heart even after amputation of 20% of their ventricle. And then this is actually the first study that was done. I think it's, yeah, 2002, maybe older than some of the people here. But essentially, as you can see, long story short, zebrafish, they have single atria, single ventricle. And then here, the figure B, they kind of cut off a little bit. And then they just fully regenerate it at day 60 if you compare to day 9. So essentially, this was kind of the first description of the cardiac regeneration in a vertebrate. That was back in 2002. And then later on, some other studies, they used lineage tracing, and then they found that all these regenerative cells, they're from the pre-existing cells. So they went through the de-differentiation and then the re-differentiation, meaning they have to go back to young and then grow old, grow more. Um, and then same thing as in the neonatal mice, as I mentioned, they still retain the regeneration, they still retain the proliferative capacity until postnatal day 7. So in the neonatal mice, they could still, if you do the same thing, if you cut the 20% of their heart, they could also still regenerate their heart. And this was also another significant finding back in 2011. And then in adult, this is the most common animal model that people use to study proliferation in adult hearts, because, okay, I'm studying a gene, I knock it out in a small little mouse and it regenerates, so what? We're not all pediatrician. So it's always more important to go back to the injury model. So this is the very commonly used injury model in the animal. We use left here. We just kind of ligate the left anterior descending aorta, and then we kind of try to create the human, the kind of mouse myocardial infarction model. just to mimic whatever happens in human. And then there are like usually two types of method. Either you permanently ligate the aorta, or you just kind of ligate for 40 minutes, 45 minutes or 30 minutes, and then you release it. Uh, Normally, anyhow, my grant reviewers always say the ischemia reperfusion is a better model that mimics human, which kind of makes sense because most of the times our patients go into the ER and then there's a PCI, percutaneous intervention, and that meaning they'll have to kind of put the stent with, or in the patients just within a few minutes. I mean, within 90 minutes. So, so yes, that's possible, but the mechanism here is different because the first one is more of a ischemia model, and the second one is, I mean, infarction, necrosis model, and the second one also involves ROS production because there's a reperfusion process. But anyhow, this is just kind of the details um, of this model. And then using this model, you can kind of study by assessing the infarction size, and then to kind of know whether your gene or your drug could help or not. And then so this is also a lesson. So now I'm gonna get into another section of what are the factors that have been studied that could influence cardiomyocyte proliferation. So the very first thing I'm talking about here is the hypoxia. So this is very interesting because I think most of the times studying the method, you can read 10 papers and you know how to study heart failure, how to do a trichrome staining, how to do a myocardial infarction surgery. But why? Like, why do you study? When you choose a project, how do you start? So this was very enlightening when I read about it. It's like when people are saying, "Oh, the kids," they say the let's say the young pups, you always need extra time to euthanize them. Whoever study mouse, they know this. It requires like 2 hours for them to get a euthanized by CO2. However, for adult mice, maybe 5 minutes is done because the younger mice, they're just very resistant to hypoxia. And then in the meantime, as we learned earlier, they also have the regenerative capacity. So are these two related? That was the initial question people asked, and then they found that actually yes, hypoxia is helpful with cardiomyocyte proliferation. And then there are just some very good papers. They prove that point. Basically, during regeneration, HIF1 alpha, which is important transcriptional factor regulating hypoxia, would be induced, and then there is a downstream cascade of cell cycle gene upregulation, and then proliferation, boom. And then the next one is another very interesting interesting study. So this one is another when people were talking, we're thinking about hormones, and then so actually this is the only drug that's so again, a lot of people study cardiomyocyte except the people here, but only one drug that could regulate cardiomyocyte proliferation is currently in clinical trials. That is the propranolol. Of course, we know propranolol is a beta blocker, and it's always used in hypertension or heart failure prophylaxis prevention, but it is just recently being used in pediatric patients, and then to see whether they could help with cardiomyocyte proliferation. So So this is one of the preclinical study, and then they found out that during development, there is an increase of beta adrenergic stimulation. However, um, if you give them beta blocker, it could actually prolong that regenerative window. Um, Again, a lesson learned from development. And then this other one is more interesting. So they kind of compared a few species, and then they found that their body temperature is negatively correlated with their oh, in fact, it's the opposite. The body temperature is actually positively negatively related to their regeneration capacity, meaning the lower the temperature, the higher the regenerative capacity. So that's when they started to use to consider, oh, maybe thyroid hormone is the one that's regulating body temperature, and that's why maybe if we intervene by giving some thyroid hormone blocker, it might be helpful to induce proliferation, and that's another kind of means to help with proliferation studies. And then I'm just kind of putting whatever that's important there. The epigenetics, because we talk about transcription, receptor ligand, and then now, of course, we talk about epigenetics. So a lot of HDAC inhibitors has been used also to induce proliferation, and then these are just kind of another way. And then sure, another type of things we try to use cell cycle regulators and then try to increase proliferation. And then this is a very elegant study. They kind of found a few combination of cell cycle regulators that could induce proliferation and help out with the MI outcomes. And then now I'm getting into, I think I only listed one signaling pathway here just because it was related to my previous study. Is this hippo pathway? I'm almost sure everybody here knows about it. Yeah, of course. So yeah, so basically hippo pathway. I think there was a fly guy here in Hopkins, and he was the one who's studying a lot of it, but now he's not here. But anyhow, the hippo pathway, when you shut it off, it would induce this kinase cascade, MST, LATS, WTR. Yep, and then they would bind to T. T was actually my project for a long time, and then they would promote uh, the cell cycle gene transcription. And then later on, there are like, I don't know, tons of paper just talking about how that if you knock out any of the component and how all of these impacting myocardial infarction, whatever that you could think of. Yeah. So this is actually our research. Again, I did not bring data, but this is a review article my student is currently writing, and we're trying to just to try to get it out as soon as possible. But this actually contains a lot of things here. I'm just gonna be on this slide for a while, but um, so essentially, currently we have a project that's kind of similar. I think it kind of touched a little bit of whatever the previous two speakers talked about. We started off with Wnt activator screening. So, oh, this is a collaboration with Chaz Hong from MSU. But anyhow, we started off with a few compounds that are supposed to be Wnt pathway activator. And then developmentally, we know that Wnt activator in the heart, they could induce proliferation, but only during the embryonic and neonatal stage. However, during adulthood, the the there are really controversial results. So what we did, we used iPSC cardiomyocytes, the 2D cultured cardiomyocytes, and then used them as a screening tool. And then we okay, maybe I'll just finish after this slide. But anyhow, so we use a few Wnt activator, and then we found a few hit, and then we kind of we found actually the story is this. We started with Dirk2 activator, which is another cell cycle activator library, and then we found one gene, one of the compounds could actually induce cardiomyocyte proliferation, and that compound was just accidentally, we found that it could induce Wnt activity, pathway activity. And then we were like, okay, maybe we should go into Wnt activation pathway. And then we found another tons of analogs from a library. And then we started to screen out which one could actually induce proliferation. So this was kind of a, a, a little bit not a very straightforward story, but anyhow, eventually we narrowed down to two of the compounds. And then we found that these compounds just worked in the cardiac organoid. So basically, these are the things we have used. We started off the screening in iPSC cardiomyocytes 2D culture, and then we go into 3D culture, and then we try, because these days in NIH, they kind of love these organoids, though they are super they're really not a very great tool, but I mean, they're like human, as they say. But with this organoid, at least we can test the function. For example, we were able to test the contractile dilatation function after treating with these compounds. And then after these, we go into zebrafish model just as another tool to see whether it could induce zebrafish cardiomyocyte proliferation. So these are the kind of the complementary tools. And then as we were writing this review article, we kind of try to look out for, okay, I was like, Daniela, this is a review article you're going to write, and 100, 1,000 grad students is going to cite your paper because they are going to want to know how to design screening schemes. So essentially, we found that lots of people they kind of started with the high throughput screening in iPSC or neonatal cardiomyocytes isolated from the mouse or rats, and then they would kind of using pretty much two types of readouts, either using a proliferation readout, for example, KX67, or a Fuji dye, and then they would continue with some validation for example, the MI model I mentioned earlier. And the other type of readout is that they would use a known target. For example, as I mentioned, the Hippo pathway or the Wnt pathway or Dirk, all these ones are the known target. They would start to design, as with the previous two speakers did, they would start to design the molecules that could target all these known molecules. So these are the two types of readouts that we eventually found out. And then, so pretty much that's how people study drugs. Yeah, I think I'm gonna just stop here because yeah. I have some more, but they are honestly the next half of this talk. I kind of just used a review article as my resource of reference. But basically, these are just trying to compare the difference between neonates and adults, and also the comparison between heart failure and normal hearts, the differences of fuel utilization between all these different models. But these are very well known. Some of them are controversial. For example, heart failure with reduced ejection fraction, that's kind of well known. But for the ones that are related to obesity or diabetes, the half-path heart failure with preserved ejection fraction, it's really controversial. I think part of the reason is that they still do not have a great animal model to mimic half-path. Um, well, I think I'm just gonna stop here.